Good morning, children. Last class, we have seen that how the new trait is obtained when two plants having different traits have crossbreed each other. Yesterday, you have seen in detail how these terminal flowers and axillary flowers are formed. Today's session, we are dealing with same monohybrid cross only, but about the other two traits, that is tallness and shortness. So 7 different features or characters I told you about pea plant. Today's session we are dealing with the tall and dwarf. We are taking. See here we are taking a pure plant and the self-pollinator. Two pure pea plants. You can see with the capital T. Capital T. This is tall. And when it is crossbreed with a dwarf plant with the genes TT, that is small t, this is a dwarf plant. How the new variety will come, let us see. This is a tall plant, this is a dwarf plant. Here there is only one, so this is in the F1 generation. You can see there is only one possibility, only capital T here. Capital T, the other capital T will be there, but at the same time, so I am showing only one. Like that, here what is the gametes formed here? Yes, it is small t, only one possibility, that is small t. So when these two fertilization takes place, you can see that a plant with genes capital T, small t is formed. Now, can you tell me what is the appearance of this plant? What is the feature it will show? Yes, it's because capital T is present here. This is the dominant gene. This will suppress this dwarf gene small t. So, the plant, when phenotypically or externally we can say that this plant will be tall in nature. Again in the F2 generation, what we are doing, we are cross-pollinating this capital T, small t into capital T, small t. What are the gametes, possible gametes formed here? One is capital T, this is F2 generation, and one is capital T, another one is small t, and here also one capital T and another one small t. So these are the gametes formed here. Now let us see. This capital T combines with this T. So I will show you in the checkerboard. As I have shown you, same way. This is a male parent. This is a female parent. And here you can see that capital T, small t. Here capital T, small t. Here in the first column, these two, so that yesterday how we have seen axillary and terminal, same way. But the letters are, we are mentioning this tall with the capital letter. And so here capital T here, this T and this T, capital T, small t. Again here, this T and this T, capital T, small t. And the last one, it is small t. So now tell me, here in the ratio of this plant, what is the nature of this plant? Yes, it is tall. Here, though capital T and small t is present, capital T will dominate the recessive small t. So, how it appears? Yes, it's a tall plant. Here, again you can see that here it's a capital T and here it is a small t. This is a small, tall plant, and here small t, this is a dwarf plant. So you can see that this phenotypic ratio, externally how we are seeing, that ratio is 3:1. That is 3 tall is to dwarf. And what is the genotypic ratio or gene ratio? Let us see. Here only there is one capital T. We see, look at these 4 columns. Only one. So 1:, how many capital T is small t? Yes, here one, here another one. So 2:1. Again here, small t, dwarf plant, that is 1. So, here you can see the phenotypic ratio is 3:1, whereas here genotypic ratio is 1:2:1. So, now we can go to another experiment with crossing the plant with the round seed and wrinkled seed. That is another one character I told you yesterday. The shape of the pea seed, one is round in shape like this. Another one you can say that it is somewhat wrinkled in shape. Now a plant we are crossing with a pure round shaped seed plant with a wrinkled seed plant. Both are homozygous and pure plant. We can see that here self-pollination takes place. Here round seeded one. We are showing with capital R, capital R. Okay, we are crossing it with a wrinkled one. We are showing with same smaller, smaller. So in the F1 generation, as you see in the tallness, darkness, same case here, gamete is only one type, that is capital R, and whereas here, there is only possibility of one type of gametes, that is small r. Okay, when these two fertilize together, you are getting a variety with the capital R, small r. Now tell me, what is the nature of this seed? Whether it is round or it is wrinkled? Yes, it is round one. Why it is round one? Because you can see that this capital R, which is the dominant character, that one will suppress this small r, that is wrinkled one. So this plant is having round seeded one. Okay. Now again in the F2 generation, here we are pollinating this capital R small r into capital R small r. Both these plants are tall in nature. Rounds, both are having round seeded one. And here the two possibility of this gametes formation, one is capital R, another one is small r. Here also in the same way capital R and small r. And when we go to a checkerboard, same thing just like the previous case, you can see that here this is the male, this is the female. Here you can see capital R, small r, here capital R, small r. So you can do by yourself, it is very easy now. The first column what will come? Yes, capital R, and here in the second column, capital R, small r. In the third one, again this r and this r. So capital R, small r. And in the fourth one, it is small r, small r. So here one capital R, capital R. So this is a pure variety having round seeded. So here also another round seeded one, but it is not pure because you can see the two types of genes or gametes are present here. You can see capital R and small r, but in appearance you can see it shows round seed. Here also this is round seeded, whereas this one is wrinkled one. So here you can see that externally your phenotypic ratio is 3:1, and again genotype, here one only, here two, and here another one. So genotypic ratio is 1:2:1. Hope that all of you understood all these things. So from all these above cases, monohybrid ratios are same. So what is meant by monohybrid ratio once more? Yesterday I told you that the kind of ratio obtained by crossing two different traits, that is with a single character. So a plant having two different traits, and we are crossing the plant and we are observing only one character, that is called as what? Monohybrid ratio. So yesterday onwards we have seen that terminal flowers and axillary flowers. tallness and dwarfness, round-seeded and wrinkled-seeded. Like the three cases we have already seen in the same with pea plant. Now from all these above experiment, you can see some of the generalized principles we can observe from all these three experiments I have shown now. You can see the first one is that each pair of the contrasting characters depend on a pair of genes. So here you can see this is a pair of genes. This is a pair of genes. See the contrasting characters. Here it is round. Here it is wrinkled one. Each one depend on a pair of contrasting characters. Each individual carries a gene in duplicate. So you can see that these genes always they will be together, and an individual produce the gametes which have only one member of a pair of genes. So here you can see here this individual produce one gamete. See it is separating. Here another gamete is there because as it is common I have not shown. So each gamete here you see it is separating here. Each one will be separating like this, and fertilization of the gametes restore the duplicate condition. Here you can see these gametes are separate in single. When they fertilize together, here capital R, small r. So duplicate condition is coming in the next one, and sex cells with respect to the genes only they contain get fertilized at random. So here you can see that R combines with this R and form here this RR. So don't think that this R will combine with this R only. So gametes it will combine in random. No, specifically this will combine with this one only. No such rules are there. It may combine in random. So that's all about monohybrid ratio. Now let us see about dihybrid ratio. So what is meant by dihybrid? Monohybrid ratio means crossing two plants having two different. We are taking only one character we are taking. Here dihybrid ratio gives us the breeding result with two pairs of contrasting characters. Together we are taking two pairs of contrasting characters and then crossing between these two plants. That is what is called as dihybrid cross, and the ratio obtained after this dihybrid cross is called as dihybrid ratio. Now you can see two characters from the same pea plant we are taking. One is with the round yellow seed and another one with wrinkled green colored seed. So the two characters one we are taking is round yellow into wrinkled green. So as you know, I got this round green first. Which one will round? Which one will be the dominant one? Yes, it is round yellow. Whereas the second one, wrinkled green, that will be the recessive one. We will represent round capital R, and yellow capital Y. Okay, and this one, so you know. Here round wrinkled, so small r, so that automatically for green, small y. Okay, so what is the possible gametes formed here in the F1 generation? Here capital R, capital Y. There is no other possibility. It is capital R, capital Y. Here it is small r, small y. Okay, when these two fertilization takes place, capital R, small r, capital Y, small y. What is the type of plant formed here? Here capital R, small r. So always this is the dominant. Capital R is the dominant one, that is round, and along with that a recessive one is shown here, that is wrinkled one. So always this will be round, and what color? Here capital Y is present, which is the super ruling or dominant one. So here you can see that along with that the green is present, that is small y. So automatically this plant will be brownish yellow. Got it? So round yellow. Again in the F2 generation, we are closing this round yellow into round yellow. That is capital R, small r, capital Y, small y into capital R, small r, capital Y, small y. Two characters, because this is dihybrid cross, we are taking two characters. So, what is the possible image formed here? You can see that one capital R, capital Y. Okay. Then next one, this one capital R, capital Y. Capital R, small y. Okay. Then another possibility, small r, capital Y. Next possibility, small r, small y. Here also the same thing, capital R, capital Y. Then capital R, small y, small r, capital Y. And next one, small r, small y. Understood how the gametes are forming in the F2 generation? So these are the gametes that are formed in the F2 generation. Now we can show in a checkerboard. So this is the male, this is the female one. Okay. Now let us see. First one, male, you can write capital R, capital Y. Then capital R, small y. Next one, small r, capital Y, and small r, small y. Again in the female section, we can write capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Okay. Now let us see how we can fill this column. Here the possibility, here capital R, capital Y, capital Y. So here capital R, capital Y, capital Y. Second, this and this, capital R, here capital Y, small y. Next, this one and this one, capital R, small r, here capital Y, capital Y. Here again this one and this column, capital R, small r, capital Y, small y. Got it? The same way how you done in the monohybrid clause. Here two characters together, that is the only one difference. Now let us see, coming to the second row. Here capital R, capital Y, small y. Second one, capital R, small y, small y. Then capital R, small r, capital Y, small y. Next one, capital R, small r, then small y, small y. Okay. Third row, here capital R, small r, capital Y. Here capital R, small r, capital Y, small y. Here, third one, small r, capital Y, capital Y. Next, last, small r, capital Y, small y. Got it? Coming to the last row, capital R, small r, capital Y, small y. Second column, capital R, then small y, small y. Third one, small r, capital Y, small y. Coming to the last one, small r, small y, small y. So you got the 16 columns also completed. Now come on, tell me how the nature of these plants. First one, capital R, capital Y. It is, you can see that round yellow seed. How we can count and write it here? Round yellow, how many we are getting? I am putting tick mark. This is a round yellow. 1. This one capital R, capital Y, small y. So here small y is present, but the dominant one is present. This is also what type of plant? Yes, round yellow in nature. Okay. Coming to the third one, though small r is present, capital R will dominate this gene, and so this is also a round yellow. Plus 1. What about this one? The both are present, but capital R is present, so it is round. Here also capital Y is present, so it is yellow. So plus 1. Here capital R. So now the chance it is round. Capital Y, small y. Yes. Now the chance it is. Okay. Then here you see it is capital R, it is round in nature, whereas here both small y is given. So what is the chance? Round green. See? Round green. I am putting an into mark here. Okay, so you are getting another variety. New variety you are getting round green. That is one you got. Okay. So here, next one. Here, capital R, capital Y, small y. So automatically this is tick mark. So that is round yellow. Then here, capital R, of course it is round one. Whereas here it is small r. So this is green one. So round green. Again here in this one we are getting. So I am putting an into mark here. That is round green one. Okay. So coming to the third row, you can see that capital R, capital Y, small y as usual. There is a tick mark, that is round yellow. Okay. Here capital R, capital Y, small y. This is also round yellow. Here. Okay. Here small r, capital Y. Small r means, yes, that is wrinkled. Here capital Y. Here yellow, wrinkled yellow. You are getting a new variety. Okay, so wrinkled yellow. Just a round mark I am putting for this one, just to identify. I am putting a round mark, wrinkled yellow. And again here you can see that small r, so it will be wrinkled one. And here capital Y, small y, that is, you can see that yellow one. So here what you got? Wrinkled yellow. Wrinkled yellow plus 1. So round mark. So, this one with the round mark, this one with the tick mark, this one with the into mark. Okay. Then coming to the last row, let us see. Here capital R, small r, so it is round only. Capital Y, small y, so it is yellow only. So round yellow. So what symbol? Tick mark. Okay. Again here, capital R, small r. It is round in nature, and small y, that is green in nature. So round green plus 1. So what symbol you have to put? Into mark. Now small r, what will be? Automatically that is a wrinkled 1, and capital Y, small y. So that's a yellow one. So wrinkled yellow here. So what mark we have to put? This one. Okay. So last one you can see small r, small y, small r, wrinkled 1, small y, that is green one. Okay. I am putting a star mark for this one. So wrinkled green. How you got? 1. So the genotypic ratio you see here. Now we can tick. How many tick marks are here? 1, 2, 3, 4, 5, 6, 7, 8, 9. No more. 9. See. 1, 2, 3, 4, 5, 6, 7, 8, 9. So round yellow you can see that it is 9 in number. Whereas round green, I put into mark. See here 1 into mark, another 2, and see the third one. See here round green, how many we are getting? Yes, it is 3 we are getting. Like that, a wrinkled yellow, put in a round mark. Here 1, 2. Here this is the third one. So 3. 1, 2, 3. Okay. Equal to 3. And last one, wrinkled green, I put in star mark. That is the last one here in this column. We can see that is 1. So equal to 1. So here you can see the phenotypic ratio. It is 9 is to 3 is to 1. This is the phenotypic ratio externally. 9 round yellow seed will be forming. 3 round green seed will be forming. Another 3 wrinkled yellow seed will be forming. And last one wrinkled green in the F2 generation. Whereas if you are looking at the genotypic ratio, it is quite difficult. There is lot of differences you can see. So in this lesson, only the dihybrid cross you have to study only about what? Yes, only phenotypic ratio in a dihybrid cross. The phenotypic ratio will be 9 is to 3 is to 3 is to 1. Hope all of you understood this one. Any doubt in this portion? Okay, so how this in the second one 9 is to 3 is to 3 is to 1 ratio is getting. From this all these experiments, certain you can see Mendel's generalization ratio. Results of these experiments are summarized under 3 laws. This who is called as the father of genetics? Yes, it is Gregor John Mendel, and he summarized based on these experiments. He summarized 3 laws, and first law is called as law of dominance. So law of dominance, already you know that when two contrasting pairs are coming together, always one will dominate the other. Here in this case, the previous case you have seen axillary and terminal flowers. Which is the dominant one? Yes, it is auxiliary. Then again you have taken the example tall and dwarf. There also tall is the dominant one and dwarf is the recessive one. Here also you have seen that the yellow and green. Yellow is the dominant, green is the recessive one. So out of these experiments, the first law is called as law of dominance, and out of a pair of contrasting characters present together. So when two contrasting, say for example, capital T, small t, two contrasting characters present together. What type of plant is this one? Yes, of course, it's a tall plant. So, when these two pairs of contrasting characters present together, what will happen? Always one will express itself, whereas the other remains suppressive. So, when these two contrasting characters present together, one will express and the other one remains suppressive. Here the T, capital T, when we are looking tall plant, this is a tall plant. This will, you can see that this will express the character what we are seeing in the F1 generation is not only, though this gamete that is small t is present. We cannot see the dwarf plant in the F1 generation. Why? Because this is suppressing or this is dominating this character. That is the first law of Mendel, that is law of dominance. When two characters or contrasting characters are present together, the only one will express itself, whereas the other remain recessive. So, second law is called as law of segregation. Law of segregation. What is the law of segregation? The two members of a pair of factors separate during the formation of gametes. So you can see that these two factors, they remain together, isn't it? But at the time of gamete formation, so you have seen this tall plant into another capital T, small t, tall plant. They are pollinating this one, and when these gametes are forming, they will separate. Here capital T will separate and small t will separate. So they will separate and each one will show its own character. So that is the second segregation, means separating. So once more, the two members of a pair of factors separate during the formation of gametes. That is the second law. And third one is law of independent assortment. So third law is law of independent assortment. When there are two pairs of characters, the distribution of the alleles means alternative forms of the genes. The distribution of these alternative forms of the character, one character in the gametes is independent to the distribution of the alleles of the other character. So you can see that this will be independent, this will be independent. You can see when the distribution of these gametes, both will be independent to each other. These are the three main important laws of Gregor Mendel, and he is called as the father of genetics. First one is the law of dominance, second one is the law of segregation, and the third one is law of independent assortment. Got it? And however we can apply this Mendel's law, application of Mendel's law. A knowledge of the basis of the Mendelian principle give us the idea about the new combination in the progeny of hybrids and enables us to predict their frequency. So you can see that by applying these laws, that is law of dominant, segregation as well as the independent assortment, we can predict what may be the new offspring, what character it will be showing. And it has a great importance not only in plant breeding as well as in the animal breeding also. You can see that these laws or these principles has great importance. And the new types of plants with a new combination of useful characters can be produced by hybridization. By applying this law, you can see that new varieties of plants or new varieties of animals we can produce. That's all about genetics. Again, yes, two small topic is there. One is called as mutation and how this mutation is caused. So, what is mean by mutation? New term, that is mutation. So how we can define this mutation? The sudden changes in the genetic material of an organism is called as mutation. So very quick or very sudden changes in the genetic material of an organism is called as mutation. So what is the genetic material of an organism? Yes, that is genes. So very sudden changes. Usually you can see sometime the children, polydactylity, here you can see usually you have 5 fingers, right? Here an extra finger growth you can see in some children. That is due to the some sudden changes in the gene it happened. Both the parents may not have this one, polydactylity. So the sudden changes in the genes, that is what is called as mutation. And you can see mutation alters the hereditary material of an organism's cells and result in the change of certain characters or certain traits. So, in the new offspring, all on a sudden a new character is found, some physical features, a new form is formed due to mutation. And for example, two examples are given here. And first one example is sickle cell anemia. What is meant by sickle cell anemia? Here, red blood cells or RBC in a to harvest that one. What is the shape it is? You can see this is the shape of the sickle. Okay, it is curved like this. This is what is called as sickle. So, the red blood cell get this shape, sickle cell shape, and the cells die very fastly. That is called as sickle cell anemia, and this sickle cell anemia is got by sudden mutation in the genetic material of an organism. Second one, due to radioactive radiations. So you again it will alters the gene structure and their effect can be seen in generation after generation. You can see that the radioactive radiation in the thermal radioactive power plant and all, you can see that this waste will be, it is deposited in very good steel vessels and deep inside the sea it is disposing. Why? Because there is every possibility of this exposure of this radiation, which is very harmful to all the living organs and not only human being, even the other animals also. It is very harmful, these radioactive radiations. So these radioactive radiation, it can suddenly change our genes. So a new, you can see that a mutation happens and new form or in any of the organ, deformity in organ may takes place. You have seen that during the Second World War 1945, that in Japan, you can see especially in Hiroshima and Nagasaki, that atom bomb was, they put an atom bomb, and you can see even now also, nearly how many years are over, so many years, nearly 75 years are over, but now also those people, those who are living in that Hiroshima and Nagasaki in Japan, they are suffering from some mutation, some deformities are forming in their body. So you can see the effect. It is not radioactive effect. It will not be completed in 1 year or 2 year or 10 year. That will be passing from generation to generation. The gene mutation will takes place. With this lesson ends. If you have any doubts, you can clarify. Just before that, you can go to the fill up the blanks and multiple choice questions and try to find out all the small answers from the textbook. Okay, thank you children.